recording going, just so I don't forget. And then I'm going to show us where we're headed today for our weekend. Awesome. All right. So today we are in our third week of our review, if you can believe it. We have one more week after this one, which will be focused on physiology and pathology high yields. So I'll be next weekend, hopefully in person, I'm hoping for. And so then we'll be done. Then it's just your own self-review until your exam that following week. So it's really, we're gearing up towards the end. You should be transitioning into mainly high yield study at this time. That's your goal. So you're looking at practice questions, flashcards, practice exams, those type of content. And for today, we're going to start with just some basic microbiology this morning. That'll be our first little mini lecture. And then we go into bacteriology. So we're going to be talking about all of our bacteria of medical importance, as well as some important components about those bacteria. So that will be what we do before our lunch break. Will be micro and bacteria. Then we'll take lunch from 12 to 1. We'll break every hour like normal. So every hour we'll take a 10-minute break. When we come back from lunch, we're going to go into virology. We're going to focus on all the major viruses that you could potentially be tested on, as well as some interesting components in regards to their diseases, their pathologies, et cetera, And then the last real 2 hours, maybe even 2.5, depending on how long we go, is going to be a deep dive into immunology, the high yield content there. Dispersed throughout, I have little practice questions and multiple choice questions that we'll kind of go throughout the presentation. It comes up a lot in our bacteria and our virus presentation, just to keep us engaged, to keep us going. So I encourage you to participate as much as you feel comfortable. And what I also have posted here, this was the practice exam we took last night and the exam answer key. I also posted some additional microimmuno practice exams if you're interested in doing more practice tests. These are focused for microbiology and immunology specifically, and the answers are also in this folder. I also have some mini review cases that I posted here. These are like couple question vignettes. So the cases are these PDFs, and then the answer key is here for all of the cases. So I encourage you to go through them. They often connect some pathologies. Some of them are multiple choice. Some of them are long answer, long form answer. It's another great way to practice. And then I have our lectures, and I try to label them. So this will be the order that we follow, the order that we go through. The last thing I posted is my micro study guide. Really, if you're trying to hone in, let's say that you've already gone through microbes a couple times and you're like, what do I need to focus in on further? This has the major kind of gram-positive, gram-negative cocci and some important components about it. It also contains your bacilli of medical importance and some important components about it. Key terms that are involved there. And so you can kind of see that this is obviously way larger than you would ever want to go through any given day. But if you're trying to hone in on, let's say, your treponema, what are the things I really need to know about treponema? This gives you kind of an outline of the types, the clinical manifestations, the conditions associated to help you really hone in your study. And I believe I also included, so you have fungi in here, and I also have parasites. So if you're wanting to expand out, the last multiple pages are all your viruses, which we will go through today. So this is just a comprehensive guide to all the microorganisms that could potentially show up on an NPLEX-like exam. They all will not, but I encourage you to just look through that as you would like. We're not going to go step by step through that guide, but it can be a helpful tool to study from as you continue forward in all of your micro study. So before we kick it off, any questions about kind of where we're going or what's on the Moodle page before we get going today?
Not about anything here. It's kind of silly too, but do you know how many options we get for NPLEX, like our multiple choice? Is it 4 or 5?
4.
4. Okay. That's what I
Yep.
thought originally, but a lot of them have had 5, so I was just trying to remember correctly. Thank you.
You will always have 4 options on NPLEX. You'll never see 5 options. So it'll always be 4. So if you can
Awesome. Thank you so much.
eliminate, if you can eliminate two, you have a 50-50 shot. So that's always the goal.
Yeah, that's where I've like been kind of working and working to be able to narrow down to at least the last two
Great.
options. So I just wanted to make sure I was like on the right track there.
Yeah, absolutely. That's perfect. That's a great strategy, especially for questions that you kind of know, but maybe don't 100% know. That's a great strategy to employ. Awesome. Great. All right. Well, oh, go ahead.
I was wondering if you have any resources for like test taking a strategy, any videos that you know, because that's one of my weakness at least.
Yeah. The first kind of intro lecture, I don't know if I gave it to this class, but I know I've given it in the past, kind of goes through my test taking strategies for NPLEX. Outside of that, oh gosh, it's past NPLEX, I believe, has an intro lecture that I believe goes through test taking strategies as well. Outside of that, I don't have anything off the top of my head. I'm sure we could find some resources together. So I'll see if I can find some during lunch and then maybe come back with some options for you.
Great. Thank you so much.
Yeah, of course, Gigi. Awesome. Perfect. All right. We'll get into it now. So I'm opening up our first lecture. This is a nice overview. I'm going to close down the faces just so they're out of the way. But this really gets into more of the kind of tests that you need to know, the media, those questions that come up on NPLEX often. So it'll be a nice, good overview of some of the microbiology concepts. A helpful way to study for micro, I find, is to create a dichotomous key. A dichotomous key is essentially your own guide that kind of helps you differentiate bacteria and even viruses from one another. And so I encourage you to do this as we go through today. We're going to approach and learn lots of different bacteria, lots of different viruses, lots of different bugs. So if you take a piece of paper, you can separate them by gram positives or gram negatives. You could separate them by morphology. Are they bacilli? Are they cocci? And from there, create a flow chart that helps you separate out your different bacteria and microbes that you're trying to identify or trying to hone in on. Your goal is with your flow chart that one bacteria is at the end of each pathway. So you start with gram positives and you create pathways that are essentially differentiating all of your bacteria in that gram positive list down to individual names. And it can be a great way, it's what microbiologists use to identify unknowns in the lab, but it can be a great study technique for you because it's very visual. It's almost like a mind map for your microbes. So as we go through this presentation, not so much, but as we go through our bacteria presentations after this, presentation 2 and 3, I encourage you to start to try to create what could be a dichotomous key or things that you might want to include in your own dichotomous key, essentially your guide to those microorganisms. These are all the different kinds of things you could potentially consider. So we talked about morphology, so different shapes of our bacteria. These are things that we should definitely know for NPLEX exam. Characteristics like do they have an endospore capability? Do they have a capsule? Do they have a special type of stain like acid fast? Are they motile? Their oxygenation or oxidation status their oxidation status. So are they aerobic? Are they anaerobic? Are they facultative? And then special tests as well. Really, catalase is the most important one here, but coagulase can come up, oxidase and urease. I wouldn't worry about the others. Hemolysis, this really matters for our strep species. Specific mediums don't tend to be tested on too often. There'll be a few that I'll bring up. Antibiotic susceptibility, typically not on NPLEX I, more on NPLEX II. Same with fermentation and sugar and temperature conditions. Those are more lower yield. But these are all the different ways that microbiologists separate out bacteria. And so as we go through our list today, you'll find that I'll highlight some of these different characteristics, and those would be helpful distinguishing categories for those microorganisms on an exam setting. Okay. So let's talk about structure of bacteria. There's there's multiple appendages, but these are the two that I want to talk about. One is flagellum, and it's made up of proteins, and its function is motility. So if a bacteria has a flagellum or a flagella, it's likely motile. Then you have your pilus or your fimbria. We can see your pili, your fimbria. These are glycoproteins, and they're really helpful for adherence. That's their goal. They also can form, the pilus or the pili can form a sex pilus, during conjugation, which is essentially the way the bacteria reproduce, the transfer of DNA from one bacteria to another. So it's helpful. Think of both Fimbria and your pili or your pilus as your adherence components, appendages on your bacteria, helping them adhere to spaces. I often think about E. coli in the bladder surface. It does a really good job of trying to adhere there using Fimbria, and then we try to flush it out by drinking lots of water to prevent getting a UTI. So that's really the example I think of the most. When we look then at some other structures, we look at our cellular envelope for bacteria. Not all bacteria have a capsule. We'll look at capsules in a moment a little bit closer, but the ones that do, they're made up of polysaccharides, the capsule, and their purpose, their function is to protect against phagocytosis, to protect against macrophages and other immune cells that can phagocytose or essentially engulf and then chemically digest those organisms. So it helps prevent identification. You also could see a slime layer. Slime layer is an S layer. This is made up of polysaccharides, and this is more of a loose network versus the capsid is more of a tight knit network. But the slime layer, it doesn't necessarily have as well-defined borders. And this helps as well as Fimbria with cell adhesion, and it can also support the formation of a biofilm. We can see bacteria that can form slime layers and even biofilm. They're common in indwelling catheters. So we'll see a couple lists here coming up that give an example of those type of microbes. So again, our capsule. So this is a helpful mnemonic for the encapsulated bacteria that have this ability to evade essentially phagocytosis. And this is please shine my skis. So the P being Pseudomonas, the S being Strep pneumoniae, the H being H influenza, the N being Neisseria meningitis, the E being E. coli, and then the SKI or the SKS is the Salmonella, Klebsiella, and Group B strep. This is a pretty high yield list to memorize, and all these bacteria are relatively important medically. So if you were gonna, there's some mnemonics I'll give you that I'll say are probably lower yield that I wouldn't worry about, but this one is pretty good for encapsulated bacteria. And so how a question could come up is if you're giving a bacteria or a microorganism to identify, and one of the tips they give you is it has a capsule, you can now rule it out to rule it into these essentially what, eight bacteria, 8 microorganisms that could potentially be your source. So again, please shine my skis is here. So how do these bacteria then get identified? They have to essentially be optimized or opsonized first, and then they can be cleared by the spleen. They can't just be directly phagocytosed. And there's 3 bacteria in this group that can also evade opsonization, and that's N. meningitis, Strep pneumoniae, which is here, and H. flu. So these 3 here can evade this opsonization, which is why these 3, there's vaccines for them, because these are typically tricky bacteria to actually eliminate if you do get an infection. And so that's why we create a vaccine for specifically these so that we can have some underlying immunity. Now looking at our actual cell envelope, remember bacteria are prokaryotes, and so they do have this cell wall, this cellular envelope, this outer membrane that's unique. And this is where we start talking about gram-negative versus gram-positive bacteria. And we'll have multiple slides that kind of goes through the what's and the whys about who's gram-negative and who's gram-positive and why they're designated as such. So this is an example of a gram-negative bacterial cell wall. So this image is gram-negative. What we can see is an outer leaflet that will typically in gram-negatives contain an endotoxin, like we can see here, LPS, lipopolysaccharides. And we'll mention endotoxins and exotoxins. We'll define them here in a moment. The lipid A portion of this lipopolysaccharide induces an inflammatory response. It induces tumor necrosis factor and interleukin-1 in our immune system. So the lipid A portion of this lipopolysaccharide is what really is triggering that big inflammatory response for a gram-negative bacteria. You'll also see in the cell wall embedded proteins. These can be porins like channels or other membrane proteins. Porins help substances be moved from one side of the surface of the membrane to the other. In the inner leaflet, you're mainly looking at phospholipids. And the keynote of our gram-negative bacteria is we have these kind of 2 spaces here, an outer membrane a peptoglycan space, and an inner membrane. And we'll look at what the difference between our gram-positive is here in a second. If we look here, you can see actually a great example. So this is our gram-positive bacteria, and this is our gram-negative. So you can see with our gram-negative bacteria, we have that outer membrane. This is where your lipopolysaccharide endotoxin would be hanging out. We have this peptoglycan cell wall, which is in between the outer membrane and the inner membrane. You have your inner membrane, and then you have inside of the cell. Versus gram-positive, you have this much larger peptoglycan cell wall and your inner plasma membrane. You don't have an outer membrane on top of this large peptoglycan cell wall. And this is partially for why gram-positive bacteria take up certain stains better than gram-negative. And that's why we can do this stain procedure, and we'll call it the gram stain, and it'll separate out these two bacteria. That's why it's such a helpful stain. So the periplasm is the space between the cytoplasmic membrane and the outer membrane. And this is where substances accumulate that are trying to exit bacteria. So we see like hydrolytic enzymes and things of that nature. And you'll see peptoglycan a little tiny bit in between this periplasm space. We only see this in gram-negative bacteria, again, because we don't have two membranes in gram-positive bacteria. And then finally, we have our cell wall, which I've mentioned, that's that peptidoglycan cell wall. It's a sugar backbone with peptide side chains. And so they're cross-linked to make a pretty strong membrane that helps the cell retain its shape. It provides that rigid support. It also can protect against osmotic changes, so it's not as sensitive to osmosis, so hypo or hypertonic solutions as cells that don't contain cell walls. And typically this cell wall portion is much larger in a gram-positive bacteria because there's no outer membrane. It's much smaller in a gram-negative bacteria. And then the cell envelope, the final, most innermost component of a bacterium, this could be considered just a typical cytoplasmic membrane. It's a phospholipid bilayer sac, and it has embedded proteins. This is where you'll find things like penicillin-binding proteins that can help with cell wall synthesis, lipoteichoic acids, which are seen in gram-positives only. These extend from the membrane all the way out to the outer exterior, and these can also essentially induce an inflammatory response. And this is also where you'll see the site of oxidative and transport enzymes. When we look at a few specialized structures before we get into more about staining, we can see the structure spores. And you can see the spores here in this image. They're the green little circles hanging off the red bacteria. Okay, so that's what you're looking at here. Not all bacteria can form spores, and so it's important to recognize that only certain bacteria are spore formers. We'll look at our spore forming and non-spore forming bacteria when we get to our bacteria lectures here later on this morning. But a spore is unique because it has a really tough coat. It's keratin-like, and it contains peptidoglycan as well as DNA. We only see spores in gram-positive bacteria. And I'd actually even go further to say we only see spores in certain gram-positive bacilli bacteria. So it's a very specialized group of bacteria that can form a spore. You could think about it as the bacteria goes into suspended animation anytime there is states that it's not beneficial to divide in. So if it's dehydrated, if it's too hot, if there's a chemical around, if there's not enough food or nutrition, these bacteria that can form spores can essentially move from their active dividing state into this spore state that then can become reactivated, almost like a seed germinating when the correct conditions arise. And so we can look and see this kind of spore-like structure here. So there's a keratin coat surrounding. This is that tough keratin coat. We only see it in gram-positive bacteria. It can survive in harsh conditions. I wouldn't worry about knowing the exact ins and outs of all of these layers. I really just know that the outer coat is keratin-like and it's very tough. And then inner core is where you'll find your DNA, your ribosomes, any enzymes that are needed to kind of reactivate and recreate the bacteria once conditions have improved. And some examples we'll see is the groupings Bacillus and the groupings Clostridium. We'll talk about all these types of bacteria here coming up, but these are the two main genus that we'll see spores in, and they're both gram-positive rods. So gram-positive being the gram stain, rod being the morphology. Looking at other specialized structures on the outer surface that bacteria can make, the biofilm producing bacteria, or bacteria that tend to have a tendency to produce biofilms, we'll see strep epidermidis, strep viridans, Pseudomonas aeruginosa, and H. Haemophilus influenza. And these are kind of the conditions that you could see biofilms being formed from, catheters or prosthetic devices, infective endocarditis, vent-associated pneumonia, and otitis media. So these are the 4 areas here. And I said strep, I meant staph epidermidis, I apologize. So staph epidermidis, strep viridans. Those are the 2 here. So now we get into the fun, the stains and growth patterns. So this is the little bit of lab microbiology that you need to know for your NPLEX. So a positive versus a negative stain. Positive stains are basic. The bacteria are negative charged because they have lots of proteins. And so they're going to take up a positive stain. So when you look at it under a microscope, you'll see blue, and blue or green. Typically, it's more blue though. And that is the bacteria taking up this positive stain. This is helpful for morphology identification. So what is the structure of the bacteria? A negative stain is acidic, and because bacteria are already negative charged, they're going to repel a negative stain. So when you look at the bacteria under a microscope, they're going to be white or clear, and the stain will be all the spaces around. So in regards to a positive stain, you'll see only the bacteria will take up color. The background will be just white or blank. In a negative stain, the bacteria won't take up the color. The whole background will be black or brown or kind of a dark gray color, and the bacteria will be bright white. And so you can confirm morphology in this image. But by far the most commonly tested on...
I have a question.
Oh, go ahead. Yeah.
Also, I don't think some people know that their microphone is on so we can hear them. I mean,
okay.
I didn't know mine was on also. So taking the quiz last night, so the gram stain is gram positive and gram negative, purple or pink. And then is there another staining called negative staining? Because I wasn't aware of that.
Yes, so these are all the different types of stains. So we're going to talk about gram positive and gram negative here in a moment, but there are positive stains and negative stains. So a gram stain, you could be gram positive or gram negative. That's its own stain, a gram stain. But you can also have a positive stain with this, which is using methylene blue, and a negative stain, which uses, it's nigrosine, nigrosine. And so these are different types of stains that are commonly used in the lab. So these are the important, and there's another stain we'll learn too called acid fast. So there's 4 staining types that I think are important to know. Does that help separate the 2?
Yes, I just
Awesome.
understand why I never got that part. Thank you.
Yeah, of course, absolutely. So now we'll talk about gram staining, Rena. So this will probably help kind of sink those pieces in. And when you're, if you were a microbiologist, there's not just these kind of 4 stains, right? There's thousands, hundreds of thousands of stains that we can do. But for medical purposes, we really only need to know a few of them. So we're only going to talk about the ones that are the medically most important for identification. But if you were a microbiologist, you would probably, like when I was teaching microbiology, we would learn at least 20 stains for an introductory microbiology course. So there's a lot of different stains that we're not even covering in this lecture because they're not necessarily pertinent for medical identification, at least for the purposes of NPLEX. But our gram stain is typically our first line lab test in bacterial identification because it gives us such a good category of separation between bacteria. And so the bacteria that have a thick peptidoglycan wall, protein carb layer, are going to retain purple stain, which is the first stain that we apply in a gram stain. The gram stain is essentially 4 liquids, 4 different stains that we apply to one slide. The first stain makes all of the bacteria turn purple. The second stain is going to kind of help push in the color further to make sure that the gram-positive bacteria that have picked up that purple stain hold on to that purple stain. Make sure you use iodine for that. The third stain, we actually wash everything away. So it washes away the purple stain from bacteria that don't have a thick peptidoglycan layer. So from non-gram positives or gram negatives. And then our last stain is pink or red. It's safranin. And so the bacteria that have a thin layer will stain pink and red, gram negative, from this process, this 4-step process. You don't need to know the steps, but I do think it's kind of helpful to recognize that all bacteria will stain purple first, then anything that's not gram-positive, as you go through the steps, will essentially have that purple wash away. And so our gram-negatives then can pick up that pink-red stain at the end, which is the last step. Gram-positives don't pick up the pink-red stain because they've stayed bound to the purple stain that was initially applied. So all of our gram-positives will stain purple. All of our gram-negatives will stain red or pink. It kind of depends on the color that you see when you see a gram stain. But we do have some bacteria that they don't play well with either stain. And so this is kind of an important group. There's a few that are really important, as we'll learn, because they have their own specialized stain that we'll use. But these little microbes may unfortunately lack real color, but are everywhere. So these are your bacteria that don't take up gram stain, either gram-positive or gram-negative. We'll still often classify them as gram-positive or gram-negative, but they aren't, it often is not a helpful diagnostic tool because they could stain either or they might not pick up the stain at all. So we can see this list here: Treponema, Leptospira, Mycobacteria, Mycoplasma, Ureaplasma, Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia. Obviously, some of them more common than others, but if you're ever asked a question about who wouldn't gram stain well, or what bacteria might be clear after a gram stain, this would be your list to consider. And so this is what this looks under light microscopy. So this would be a gram-negative bacterium, and it looks like gram-negative rods based off of the shape, the morphology. And this looks like a gram-positive. It looks kind of like rods, although it might be cocci just bound together. It's hard for me to see from this depth away, but you can see the distinction in color, right? This pink for gram-negative, this purple for gram-positive. And as I mentioned, bacteria with that thick peptidoglycan layer retain the crystal violet stain, which is the first stain applied. And then the thin peptidoglycan layer, your gram-negatives, they get that stain washed away in this step 3 here with this alcohol or decolorizer. And so they pick up the counter stain, which typically is safranin. That's typically the counter stain applied. Okay. So this is a list here. If you are trying to kind of create these different categories for yourself, this is a list of a lot of the bacteria we'll go through in our next group. Our next slide is about our gram-positives. So these are our gram-positive bacteria. All these bacteria will stain relatively purple under gram stain. So they have a thick peptidoglycan wall. And so that's one of their characteristics of these bacteria. Versus this is going to be your list of your major gram-negative bacteria. We'll go through a majority of these as well. These gram-negative bacteria will stain more red or pink. They don't have as thick of a peptidoglycan wall, and they do in fact have those 2 layers, right? Those 2 cell membrane layers, like we saw at the beginning when we were looking at that one slide where we saw a cell membrane layer, a thin peptidoglycan wall in the middle, and then a cell membrane layer. Again, these are not all bacteria gram stain. So helpful, trying to help you kind of identify who and why. These ones are too small, so that's why they are unable to take up gram stain. These mycobacteria has a cell wall with too much lipid, too much fat content, so it doesn't take up the stain well at all. These don't have any cell wall, so that's why they don't take up stain. And then the rest of them here are they're typically intracellular. So they're dividing inside of a cell. So their cell wall is different in nature. They don't have that peptidoglycan layer necessarily, and they're much smaller. They act somewhat like viruses in that they need to be inside of a cell to divide and to grow. The other stain that I mentioned that would be important to know would be our acid-fast stain. I've never seen it referred to as the Ziehl-Neelsen stain on NPLEX, but acid-fast stain uses a carbyl fuchsin dye. It turns the bacteria red, so the bacteria that are red are acid-fast bacteria, and Mycobacteria and Nocardia are the 2 bacteria groups that take up this stain. And this is an NPLEX question often. So recognizing acid-fast stain is associated with Mycobacteria, Nocardia. Both these bacteria typically cannot take up a gram stain. So we use this staining method instead. All right. A few other funky kind of micro lab techniques. There are different ways that we can grow bacteria. So these were ways we could stain bacteria. So on a slide, applying different dyes, but we can also grow out bacteria in the lab, and we grow them out on these different agar plates. And agar plates are typically, like our standard agar plate has some soy, some sugar, some gelatin, and that's pretty much it. Sometimes some other like molecules and salts and things to help bacteria grow. But there are specialized agar that sometimes are brought up on NPLEX, and there's so many different types. I'm only going to mention a few here because I don't want to, I don't think you should know them all, but there are a couple that can come up a little bit more often. So chocolate agar is unique because it is a blood agar, and then the blood agar is heated up to form this kind of chocolatey type color. It is the identification agar used for Haemophilus influenza. So Haemophilus influenza will be the bacteria that grows on this agar. So we'll see kind of this smooth gray colony. If you see that growth pattern on this agar, then you'll know that you have this bacteria. So it's a one-to-one, it's an identifier. That's why I think it's important. McConkey agar, we'll see it provides a fermentation result. So this is a plate that's just uninoculated, so no bacteria is on it. And then you can see when there's like this pink colony, these are fermenters of lactose. And so this is helping us see what types of sugar or what types of metabolism these bacteria have. And so with McConkey agar specifically, you'll find any bacteria that grows on it and forms this kind of pink colony is a lactose fermenting bacteria. And these are categorically our enteric gram-negatives. So the gram-negative bacteria that we find in our digestive tract. These can either be commensal bacteria that could typically live with us in harmony, or bacteria that could cause us harm and cause us gastrointestinal distress. And so we can help separate out some of these lactose fermenters or enteric gram-negatives from lactose non-fermenters like Salmonella, Shigella, and Proteus. Eosin methylene blue, EMB agar. The colonies that grow here have this really green sheen, and this is the identification agar for E. coli. So eosin methylene blue is identification agar for E. coli. And our sabrord agar is unique because it's for fungi explicitly. It's too acidic of an agar for bacteria. So you're going to only see fungi grow on this agar, and there's not many that do that. So that's why it's unique and deserves to be called out. Okay. So then looking at our metabolism of our bacteria, this is where we talk briefly about anaerobes, aerobes, and facultative. And I say briefly because we're going to talk more about this when we get into each individual bacteria here coming up. But I do have a couple mnemonics that are helpful that I wanted to place here up front. And then when we actually look at these species later on today, we can delve into some other characteristics about them as well. So anaerobes can't breathe fresh air. This would be our Clostridium, our Bacteroides, our Fusobacterium, and our Actinomyces israel. And these are bacteria that are unable to deal with oxygen. They do not have the ability to handle reactive oxygen species. They are purely anaerobic. They can only grow in places where there's no oxygen, where oxygen can't touch them. So you typically won't find these on the surface. You typically won't find these in your respiratory tract. You'll find these kind of deep inside, hiding within spaces that aren't necessarily touched by oxygen. So here you go again, can't breathe fresh air. You could see all of these potentially being in normal microbiota in your GI tract, but if they get elsewhere, they're pathogenic. As I mentioned, they're susceptible to oxidative damage because they do not have catalase or superoxide dismutase. They have a pretty distinct odor due to the small chain fatty acid content, and they can produce gas in tissues like carbon dioxide and hydrogen. So they are pretty stinky anaerobes. And then the kicker here is our anaerobes don't have catalase, they don't have superoxide dismutase, they can't deal with oxygen. Our facultative anaerobes are going to be your largest category, and they can use oxygen as a terminal electron acceptor to generate ATP, but they can also use fermentation or other oxygen-free pathways. So there's so many. A few examples are your streps, your staphs, your enteric gram-negatives. but there's way more. We'll see that this is probably the most common type of oxidative status of microbes, of bacteria. And then we'll find our aerobes. I don't have a category of them here, but I have them listed throughout. So we'll find our aerobes throughout our lecture here. A few other pieces, I have intracellular bacteria, another kind of unique metabolic component. Some bacteria are obligate intracellular bacteria, meaning they have to stay inside cells. So that's when it's really chilly and cold. So Rickettsia, chlamydia, and Coxiella, they cannot divide outside of a cell. They have to be inside of a cell to divide. Versus these are facultative. They like to be intracellular, but they can divide outside. So that's some nasty bugs may live facultatively. The ly is the Ly. So these are your facultative intracellular bacteria. They like to be inside of a cell. They have to be inside of a cell to divide. I mentioned oxygenation status. And so when we are thinking about how do these bacteria deal with reactive oxygen species in the body, and we'll look in our immunology lecture about how reactive oxygen species can be utilized in our body to actually attack bacteria and microbes. But bacteria have developed mechanisms or strategies for dealing with that. Catalase can degrade hydrogen peroxide into water and oxygen. And so any bacteria that's catalase positive, they are able to deal with oxygen and reactive oxygen species. So these would be at the minimum facultative anaerobes or aerobic, meaning that they can deal with oxygen. And so this is our big catalase positive organisms. Cats have been to places. And so that's your big is the B. And then let's see. Oh no, cats have B and then H. Oh, maybe I have the other B here. I think the first B is here. I don't know how it works. At some point, I had this acronym that made sense. I'm sure I could figure it out again. But these are your bacteria that are primarily catalase positive. When in doubt, a lot of bacteria are catalase positive. If they can deal with oxygen, if they're facultative, they probably have the ability to be catalase, or they have some other way of dealing with reactive oxygen species like oxidase positive. But Staphylococci, this is often represented as one of the big major catalase positive bacterium. And it's important to note that Staph is catalase positive, it's gram positive, and it's cocci in shape. Streptococci is gram positive as well. It's cocci in shape as well, but it's catalase negative. So our strep species and our staph species, catalase is a great way to tell them apart from one another. Genetic-wise, I don't have a ton about genetics in this intro slide. We'll kind of talk a little bit about this throughout. We'll talk more about this in our viruses. But there's 4 words I want you to understand: transformation, conjugation, transduction, and transposition. Transformation is bacteria are able to bind and import short chromosomal DNA sequences. And then below, I have some bacteria that employ that strategy. Conjugation, all bacteria can do this as long as they have a pili or pilus. A single strand of plasma DNA is transferred across a conjugal bridge. That's typically a pilus that's formed between 2 bacteria. You're able to pass plasma DNA from one bacterium to the next. Transduction is dealing with a lytic phage that's going to infect bacteria, leading to cleavage of bacterial DNA. The phage capsule can then contain bacterial DNA by accident, which can be transferred to a new cell. So this is where we start thinking about bacteriophages. Essentially they look like a virus almost, but they can transfer bacterial DNA from one bacterium to the next. And then transposition, DNA portion copies, cuts itself out, and inserts itself into a DNA molecule. And this is how we get drug resistance in our bacterium. So I don't see these often on NPLEX, but I do think it's important to kind of understand the differences between these 4 words because they're all dealing with kind of the genetic components of our bacterium. Okay. Few last moments for some special tests before we wrap up this one and then get into more of the fun where we can kind of test ourselves and our knowledge. Our catalase test, this is an example here. Catalase positive is the bubbles. Catalase negative is no bubbles. Catalase, again, degrades hydrogen peroxide to water and bubbles of oxygen, hence why a catalase positive looks like this. And this is the same, cats have been to places. Risk factor is if someone is NADP oxidase deficient, that can lead to a recurrent infection for these people because they are actually unable to deal with this catalase enzyme. They're unable to essentially disrupt this process if they don't have NADPH in the body. So we can see that we can start to kind of see how some of these pathologies can link over and deal with complications for us humans. Urease is probably less common. The only reason I mention it at all is just because I've seen it come up one time. So I was like, well, I'm gonna have an acronym for it or a mnemonic for it. And it's also a really pretty picture. The urease positive. It's this like really bright hot pink, change in color in either broth or a slant tube. But urease hydrolyzes urea to release ammonia and carbon dioxide, which is going to increase our pH. It's more basic. And so we'll see our urease positive bacteria could actually make our urine more alkaline. And so P chunks is going to be your acronym, your Proteus, ureaplasma, nocardia, cryptococcus, H. pylori, Klebsiella, saprophyticus, which is a staph species, and staph epidermidis, both a staph species. When you have a Proteus bacterial infection, you are actually predisposed to struvite kidney stones, and that they're magnesium ammonium phosphate stones. And so that is a complication of a Proteus infection. It's a struvite stone, and struvite stones are those large, really like big, awkward looking kidney stones that can be really challenging to pass on their own and often potentially need surgical removal. So it's something to be a bad complication, but something to keep in mind. Here's your urease positive organisms. And then our last but not least, we have to talk about our virulence factors. This is where we get into endo and exotoxins. So on the outside of capsules, or just a capsule in general, the bacteria that contain a capsule, it's a polysaccharide. It's highly charged, it's hydrophilic, so it plays well with water, and it essentially allows those bacteria to be barred from being phagocytosed or lysed. And so it allows them to stick around and hide out in the body longer and divide longer before being tagged as foreign and start having an immune response be mounted. Another interesting protein that can be on the outside or the surface layer of cells that's specific to Staph aureus is protein A. Protein A binds to the Fc region, which is a specific component region of IgG antibodies, and we'll see that region when we look at our immunology lecture. And again, its job is to prevent opsonization and phagocytosis. So Staph aureus, not only can it evade phagocytosis, it can prevent antibodies from even tagging it as foreign because of this protein A that can bind to the antibody itself and prevent it from identifying Staph aureus. So it's really good at hiding out. That's why a staph infection can often kind of grow and fester even before we recognize it's there. M proteins are expressed by group A strep, and they help, again, prevent phagocytosis because it looks like cardiac myosin. And so because it looks like our own self cells or our own self myosin in the body, the immune system doesn't want to bind to it because it thinks it's our own cells. This then eventually, once it does get identified as foreign, can contribute to some autoimmune responses, specifically for acute rheumatic fever, which is caused by group A strep. So we can see some complications here if the body recognizes it on group A strep, starts creating antibodies towards it, and then we kill off the group A strep. But now we have antibodies to our own cardiac myosin, and we start attacking our own cardiac myosin, even though the bacteria is gone. So bacteria become really tricky at being able to kind of evade our immune system detection and try to cloak themselves to look like humans or their host. IgA protease is an enzyme that cleaves IgA enzymes, and IgA or IgA antibodies. And IgA antibodies are often secreted for like our mucosal pathogens. We see it in our salivary region. We see it a lot in the gut being secreted. And this protease essentially breaks this down, and it allows for bacteria to adhere to and colonize more of our mucous membranes because those antibodies are unable to bind, opsonize, and tag them as foreign. So our strep pneumoniae, our Haemophilus influenza type B, and our Neisseria species are really good at being able to break down that IgA and then adhere to places, especially mucosal membranes, and start their infectious journey. All right, I promised it. So here's my delivery. Exotoxins and endotoxins, probably the thing we all get the most confused about. So exotoxins first are proteins that are produced inside a bacteria. They're most commonly seen in our gram-positive bacteria. And then they're secreted or released into the surrounding medium following lysis, following the cell breaking apart. So think of exotoxins are exiting the cell, and they're produced inside of a cell first. So they have to exit, right? So they're inside the cell where they're produced. They're an exotoxin that's going to exit. And then they're typically associated with gram-positive bacteria. Versus our endotoxins, these are the fatty, the lipid portions of lipopolysaccharides, LPS, that are part of the outer membrane of the cell wall of gram-negative bacteria. And remember, our gram-negative bacteria has, if I draw a beautiful picture, has an outer membrane, are in between peptidoglycan cell wall, and an inner membrane, and then you have the intracellular space. So the lipopolysaccharides are hanging off this outer membrane, and they're hanging out here, and it's the fatty portions that are really the toxic part. These are seen on gram-negative bacteria exclusively because gram-positive bacteria have just an inner membrane, and they have a really thick cell wall, but they don't have an outer membrane on top of it. So they don't have anywhere for an endotoxin to adhere to, okay? The endotoxins are essentially liberated or released on their own when bacteria die and the cell wall breaks apart, but they're not excreted. They're not purposely released. It's only when the bacteria die and the cell wall breaks apart that you would ever see an endotoxin floating around on its own. Otherwise, they're adhered to that outer cell membrane. So going back to our gram-positive, most likely exotoxins, although some gram-negatives can create them, so it's not exclusive here. But our exotoxins, remember, are secreted from a cell, but the genes are in the plasmid or the bacteriophage. They're created inside of the cell, secreted outward. They're polypeptide in nature, they have high toxicity levels, and they're going to induce a lot of antibodies. They're going to be essentially a big immune response. The antibodies that are created, we often call antitoxins. You can use these exotoxins as vaccines, and we see that used relatively often. And they can be destroyed at high elevated temperatures, except for staph enterotoxin and E. coli heat stable toxin. So some things we could think about is tetanus, our tetanus toxin, our botulinum toxin, diphtheria, and cholera. All of these have exotoxins that are created in the cell, secreted out of the cell, highly toxic, and a really heightened immune response that occurs.
Dr. Pesha?
Yeah.
So antitoxins and toxoids are synonymous.
Antitoxin, close, but not quite. Antitoxins are the antibodies that we create to attack the exotoxins. Toxoids are the synthetic version of the toxin that we can inject as a vaccine to then create our antitoxins.
Got it. Thank you.
Yeah, absolutely. Great question. Is that Dylan?
Yeah, that was me.
Yeah. Okay, awesome. Sorry, I couldn't see your face, but I was like, I recognize the voice.
Yeah.
Awesome.
Well done. Thank you.
Perfect. Thanks. All right. So a couple other examples here with specific mechanisms. So we can see Corneobacterium diphtheriae creates diphtheria toxin, and Pseudomonas aeruginosa creates exotoxin A. Both of these are going to inactivate elongation factor, which is essentially inhibition of protein synthesis. So they're inhibiting protein synthesis within our cells. Not so great, especially if we're trying to create antibodies and create immune cells, and we need to do a lot of replication, a lot of cell creation, differentiation. If they're able to block that process, it's going to slow our response. So we see, obviously, with diphtheria toxin, we see a pharyngitis with pseudomembranes in the throat, severe lymphadenopathy, that's LAD, and myocarditis, inflammation in the heart muscle. With exotoxin A and Pseudomonas, you can actually have cell death. Host cell death can occur. This isn't host death necessarily, but host cell death can occur from this inactivation. So it's not just a localized response, as you see with diphtheria toxin. you could see across the board cells not functioning anymore. With our Shiga toxin or Shigella and our enterohemorrhagic E. coli Shiga toxin, it also has a Shiga toxin. Both of these inactivate the 60S ribosome by removing adenine from rRNA, so also inhibiting protein synthesis, just different mechanisms of doing so. And you'll see damages to GI mucosa, so dysentery. You'll see an increased cytokine release, so hemolytic uremic syndrome. So those big inflammatory responses, these cytokine storms, so to speak, that almost harm the body more than the original bacteria did. So these toxins, that's a lot of the danger with these toxins, is the huge inflammatory response that occurs almost can overwhelm the host more than the original bacterial infection. Again, a couple other examples here. So enterotoxic E. coli, this is a heat labile toxin. We have an example, it's going to overactivate our cyclic AMP, so that G protein pathway, which will increase chlorine secretion in the gut, and it'll increase water efflux. So we see then watery diarrhea because chlorine and water are moving out into the lumen of the GI tract. And so your cholera, your anthracis, pertussis, E. coli, you can see kind of this whole picture presenting here. Bacillus anthracis is a heat stable toxin. Again, attacking that G messenger pathway, it's going to lead to decreased reabsorption of sodium chloride in water. And so again, watery diarrhea. Your cholera toxin, Vibrio cholera, overactivates again that cyclic AMP by permanently activating G proteins. So it's a permanent activation of that second messenger pathway. So you just have this constant upregulation until that cell dies. And so that's why you see this huge amount of rice water diarrhea that just can't stop, because literally you have a positive feedback mechanism unchecked in this instance. And the Bordetella pertussis toxin, cyclic AMP is activated by inactivating its inhibitory subunit, so the G protein. So it's essentially also almost permanently turning it on. And this is where you see whooping cough. So of course we can treat all these diseases. We don't just die from all of them, but you will see a pretty significant immune response from the body, often significant symptomology, and you're pretty sick for these conditions. And this is why our exotoxin is really the cause here, the causative agent. I have some more going through. We're going to talk about all these bacteria individually too. So this is not your only time seeing them. This is just kind of focusing on their exotoxin in a table just to make it easier. But our Clostridium species, both tetany and botulinum, they have proteases, things that break down proteins that cleave our snare protein receptor and are affecting essentially neurotransmission. And so we'll see those differences when we look at these pathologies. But in our tetany, we can see inhibition of inhibitory GABA and glycine neurotransmitters. So spastic paralysis is seen in our tetany. And then you can see ingestion of this toxin here. This is where you get your flaccid paralysis with Clostridium botulinum. We'll talk about this later. So this you're going to see this more again. Flaccid paralysis. Your Clostridium perfringens has alpha toxin. This is your gas gangrene hemolysis response. And your streps can create streptolysin and erythrogenic exotoxin. They can essentially break down the cell membrane. They can lyse red blood cells. This is where we get to our hemolysis that can occur from our strep species. And then Staph aureus, our toxic shock syndrome toxin. This is essentially going to cross-link the B region of our T cell receptors to our major histocompatibility complex class 2 of our antigen presenting cells. So it's all these acronyms. We're going to learn them all outside the antigen binding site. So essentially, it's going to upregulate the production of all these pro-inflammatory cytokines because it permanently locks in and kind of binds these two regions of these two different cells. And because we have an upregulation of these pro-inflammatory cytokines, we see shock as the response, because our pro-inflammatory cytokines, what are they going to do? They're going to vasodilate. They're going to increase fluid flow to that area. They're going to allow for increased diapedesis, movement of stuff from the blood to the tissue. So all of this stuff is going to lead to low blood pressure, low blood volume, because we're vasodilating, and so ultimately shock. Whew, exciting. Exotoxins, they mess shit up. That's pretty much what you can think about that. Endotoxins aren't great either. They're in their gram-negative cells and the outer cell membrane only. So they're only going to be in gram-negatives, only in that outer cell. They're not secreted. The genes are in the bacterial chromosome. And it's really the lipid A component of lipopolysaccharide of LPS that's the main causative factor here. It's lower toxicity, but it still can cause fever, shock, hypotension, even disseminated intravascular coagulation. So it's not without fault, but it's less toxic than an exotoxin, partially because an exotoxin can be released into systemic circulation easier. It will induce your pro-inflammatory cytokines, so a pro-inflammatory response. It's poorly antigenic, so it's hard for us to create a vaccine for an endotoxin, which is why sometimes our gram-negative bacteria can be more toxic, because we can't necessarily create a vaccine to target this toxoid. It's because there's not a great antigenic read or lead for us to be able to synthetically create that. And they're stable in hot heats for a while. These are some symptoms that can happen, or the responses for endotoxins. So not great, obviously. Pro-inflammatory and pro-inflammatory cytokines here. All right. Awesome. Let me get out of here and see your faces. So that was like my quick and dirty overview of microbiology. If you had like one thing that you're going to review, I would go through this, because this gives you an overview on a lot of stuff. It doesn't go into a detail on a lot of things, right? So it's going to be an overview of surface level. We're not deep diving into the bacteria yet. We're going to next. We're going to go through all of our gram positives, and then we're going to go through all of our gram negatives. That's what's coming up before lunch in the next couple hours. But this is still supposed to be like, okay, what are we looking at overall when it comes to microbiology? All right. What other questions do we have? We had some good questions during this first little chunk.
I have a question on, I'm just trying to organize it in my brain for the
Yes.
virulence factors. So does the slime layer and the LPS or are those considered virulence factors, or are they considered like a subset, or I'm just not sure how that fits in.
They would classify as virulence factors because they're helping the bacteria become more infective or cause more disease. Yep. So slime layer, biofilm, capsule, all could be considered virulence factors.
Okay, and then the flagellum and the fimbria, are those considered virulence factors too, or are they just like components?
Absolutely, absolutely. They would be, because with motility, the bacteria can get into other places easier. It can move around. It can evade the immune system. And fimbriae, it can adhere to spaces more effectively. So absolutely.
Awesome. Thank you so much.
Yeah, no, great question. Perfect. All right. Well, on that note, we're going to break, and then we come back, we're going to get into our first bacteria PowerPoint. So it'll be our bacteriology part 1. And when we get into this, you'll see we'll present kind of a chunk of bacteria, and then we'll have a section of questions that we'll kind of answer together. And so I encourage you, don't look ahead at the answers for the questions. Try to truly test yourself to see what your quick recall knowledge is. And then obviously you'll get the answers as we go through the PowerPoint. So you will get them. But try not to look ahead too far so that you don't cheat too much. All right. I will see you at, we'll do 10 after, and then we'll get started again. So 10 after 10, we'll come back together.